Hello everyone, welcome to join today's webinar. My name is Michael Zhou, the Global Product Manager for Peptide Portfolio at GenScript. It is my privilege to introduce today's speaker, Dr. Anna Passetto. Anna joined the Karolinska Institute in 2018 as Assistant Professor and the Managing Director of the Pre-GMP facility in the Department of Laboratory Medicine. She received a master's degree in molecular biotechnology in 2007 from Alma Mater Studio Bologna University, Italy. She then moved to Karolinska Institute, where she received a PhD in 2012. She joined the laboratory of Dr. Steven Rosenberg at National Cancer Institute, NIH, in 2013, and her research was focused on the isolation of TCR genes from mutation-reactive tumor-infiltrating lymphocytes in metastatic solid cancer. Today, Anna will provide us a background on the most successful approaches that led to the identification of immunogenic neoantigens for cancer treatment. We'll also discuss limitations and the potential improvement to the discovery approach. Without any further ado, I will pass the floor to Anna. Anna, whenever you are ready.
Thank you, Michael. It's really a pleasure to be here for this seminar and to have the opportunity to share, my experience on this topic. And so the title of the seminar is TCR Targets Validation through T-cell Activation. And here I prepare an outline for the talk. So first we will start with some background about what are the T-cell targets, especially in the context of cancer immunotherapy, because that's my main expertise. And also I will try to clarify the difference between mutations and neoantigens, and also to describe what happens when the T-cells encounter its specific antigen. Then we will proceed with a little bit of overview about the most common methodologies to identify T-cell targets through T-cell assays, and also some applications about TCR isolation, especially from the cancer immunotherapy field. So what are T-cell targets? In this presentation, we will only talk about alpha-beta T-cells, for simplicity, and because that's the cell type that I work with the most. But there are also other T-cell subset like gamma delta T-cells and mate cells that may function a little bit different. So when I will refer to T-cells, I mean actually alpha-beta T-cells in this presentation. So the T-cells can be divided into main subgroups: CD8 positive T-cells that usually recognize peptides within the class 1 HLA complex, and CD4 T-cells that recognize peptides in the class 2 MHC complex. And the mechanism of recognition is that the T-cell receptor on the T-cell is able to make contact both with the specific peptide and also the HLA residues. So that gives a specificity for both the peptide, so the antigen, for example, that is recognized, but also for the HLA molecule, like the HLA restriction of that specific TCRs. And CD4 and CD8 antigens usually differ from a structural point of view, because CD8 antigens or peptides are usually shorter, let's say 8 to 12 amino acids, while the CD4 restricted, or like the class 2 restricted, are longer, up to 25 mers or 12 to 18 on average. And also usually class 1 restricted antigens come from intracellular antigens. For example, if it's a virus or a self-antigen that has been processed by the intracellular mechanism of the cell. while class 2 antigens usually come from more an extracellular path, like endocytosis and so on. And in the context of immunotherapy, for example, or cancer immunotherapy, so what would be an ideal target? So now we are talking about cancer cells. So an ideal target should be present only on cancer cells. So to avoid the risk, for example, of targeting normal tissues that may express otherwise the same antigens. This antigen should also be expressed in a kind of homogeneous way on all cancer cells to avoid to, for example, target only a subgroup of cells. It should be also highly expressed so that the recognition is easy for the T cell, and also highly immunogenic so that it actually generates a potent anti-tumor response. And then when thinking about therapy, if the antigen is also shared among patients, for example, it can be a more interesting target because with the same type of therapeutic approach, one could treat several patients. And if we look at this this picture here, it represents the distribution of intracellular and intracellular potential antigens. And like membrane-associated proteins or extracellular antigens are in reality just a minority compared to all the possible intracellular protein that can become antigens for TCRs. And today we will briefly talk about neoantigens, and in particular the difference between public and private neoantigens. But before that, it's I think important to clarify what are also unique mutation compared to hotspot mutation. So what is really the connection between a mutation and then an antigen? So we can start with the mutations. So mutation is in general when something happens to the sequence of a protein, so that you have a change in the most common amino acid sequence, for example. So in this picture, this is like a gene view histogram from the website Cosmic. And I took as an example the protein P53. And so every histogram represents a position in the protein, so an amino acid position. And then the histograms, like the height of the histograms, represents how many times this position has been found with a different amino acid, or like with a substitution or a mutation. And across all the length of the protein, you can see that some histograms are much higher. And so those are called the hotspot mutation, because those are regions where it's highly frequent to find a mutation. And here I just put as example a couple of zoom in histograms where you can see, for example, in the position 175 that used to be, or like the wild type version has an arginine, and it's very frequently mutated towards an histidine. So if we think about all the unique random mutation that can happen by chance in a gene, so the hotspot mutation is like a subgroup of the potential changes that can happen. And so how do we go from the mutation to a neoantigen? So a neoantigen is when this mutation results in a protein change, and that protein change results in a peptide that is presented on the HLA of the cells, and a T cell is able to recognize it. So an antigen can only be antigen if it's recognized by a T cells in our case. And so when you have like private neoantigens, it means that these antigens are originate from random unique mutations, that they are private or unique for each patient, because you will not find two patients with the same mutation, let's say, or it will be very difficult. While shared neoantigens originate from hotspot mutations, and that those can be found in several patients. But again, like the shared neoantigen are a subgroup compared to private neoantigen. And another thing that is really important to remember is that an antigen for T cells is also associated with the HLA restriction. So it can be that two patients have the same mutation at the genetic level or at the protein level, but when it comes to HLA presentation, if they have two different HLA haplotypes, for example, then the actual peptide that will be presented will have a different sequence. And so the antigen itself, it will be different for the two different people, even if the mutation originally is the same. So an antigen always have to be in the context of a specific HLA restriction. So then what happens when the T cells reacts or when encounters its own antigen? And this means when the T cell receptor is engaged and can contact both the peptide and the MHC molecules. So different things can happen, and you can have, for example, proliferation. So the T cell starts to duplicate. It could also happen that you have cytokine secretion, and different cytokines can be secreted depending on the different T cell type or subpopulation that you have. And also in case, for example, of effector T cells, you can have directly a cytolytic effect and killing of the target cells, so the cells that present the antigen. And so now I will just briefly give some example of assays that can measure this type of reaction of T cells. So if the reaction is proliferation, for example, it is possible to measure this proliferation of this cell expansion. And the one thing to consider is that now we will talk about measuring it in vitro. So we don't know what's really happening in vivo, but these are all assays that one can do in vitro. So in the in vitro condition, you need to provide the stimulus for the T cells. And this stimulus can be TCR specific. So when you know the immunogenic peptide that you want to, so that you know basically the antigen. So you have your own target and you know the sequence of it. So you use that to stimulate specifically the cells with the TCRs that is able to recognize this peptide. Or you can also have a non-specific stimulus when you just, for example, stimulate a whole population to see if it's even capable to proliferate anymore. For example, if it's an exhausted population, you just want to see if it's still able to duplicate. And then you can use unspecific agents like anti-CD3 antibody or like anti-CD8 or PMA ionomycin, for example. And this, so one possible way of measuring the proliferation of the cells is to give a dye, to label the cells with the dye that will enter in the cells, and then measure the dilution of this dye over time. And then you will see the peaks basically of the different cells generation over time. And then you can, if you have different subset or different stimuli, different concentration, then you can compare and see which stimuli gives a more proliferation or triggers a higher proliferation rate. Then another measure of T cell activation is to see how much cytokines are released and also what type of cytokines are released. And depending on the T cells, the population, it can be a different type of cytokines that are secreted, and also the cytokines themselves can even further help to understand what kind of population has been stimulated. So very simple distinction is from effector cells like CD8 cells and CD4 cells. Usually effector cells, they secrete interferon gamma, IL-2, TNF, TNF-alpha sometimes, while helper cells, they can also secrete gamma and IL-2, but depending on different subset, they can also secrete, for example, IL-10 or IL-4, and then another specific population can secrete IL-17. And how do you measure these cytokines? Usually, or like the simplest way is to measure the concentration of this cytokine in the supernatant of the cells. So the cells are in contact with the antigenic stimulus, that could be a peptide, for example, and then they secrete the cytokines in the supernatant. And you can take the supernatant and do an ELISA, for example, an immune assay where you have antibodies that can detect the cytokines. It could also be done with the flow cytometry, where again you use antibody to detect the cytokines, but when they are still in the intracellular compartment of the cells. And you do that, for example, using some agents that will stop the secretion in the cells, and then you can permeabilize the cells, fix them, and then detect the cytokines while they're still intracellular. And another possibility to measure T cell activation is to see if there is any surface marker that is upregulated on the surface of the cells. And then depending on the length of the or like when is the time point where you want to assess the activation, then you need to choose different markers. For example, there are early activation markers where you see upregulation only after a few hours of from the initial stimulant, or you can have later activation markers that require more time. So the kinetic is a bit different between the different markers. And you can also have markers that works better for CD4, for example, or for CD8. For example, CD137 usually is expressed mostly on CD8, while CD134 or CD154 are expressed mostly on CD4. And then another way, another assay that can be done is also to measure the lytic activity, and that's usually like a killing assay, for example, with targets that are labeled with certain dyes, and then you can measure the either the disappearance of the dyes, or in the past was used a lot radioactivity to see the release of radioactivity from dead target cells. But so how do we validate the T cell antigen? So how do we say that this potential target is really an antigen? So I wanted to give the example from the cancer immunotherapy field, and especially the work at the surgery branch in Dr. Steven Rosberg laboratory, because it was one of the pioneer in the field. And the idea behind is really to measure the T cell activation after encountering a proper antigen, a proper stimuli. And the antigens that were, or like the targets that were evaluated were peptides or synthetic mini-genes that were derived from mutated sequences of protein present in the tumor. So as is described here schematically in this picture, the whole process starts with the sequencing of tumor samples. And the tumor contains a lot of mutations, and we saw that could be unique mutations or also shared mutations. But all these mutations are then transformed, let's say, into a target for T cells by having peptide, synthetic peptides, or mini-genes that encode exactly the same mutated sequence. And then these peptides, for example, are then loaded on the antigen-presenting cells of the patient, and the T cells, the TILs from the same patient, are placed in contact with these antigen-presenting cells loaded now with this mutated peptide. So as we said before, a mutated peptide can only become an antigen if it finds a T cells that it's able to recognize. So you really measure the T cell activation at the end, and then you say if the T cell was activated, this means that the mutation was able to become an antigenic peptide, so an immunogenic peptide. Therefore, we can say that that's a potential target also for immunotherapy. And the activation of the T cells can be measured in all the ways that we described earlier on. And in the surgery branch, one of the most used way was to measure the expression of surface marker like 4-1BB or OX40. And this is because having a surface marker as indicator of T cell activation also enables to do, for example, T cell sorting and then TCR isolation that then can be used for TCR T cell therapy. And here I just reported a few publications that describe how this neoantigen specific response is found in a successful adoptive cell transfer. And also here another example of what type of antigens have been found, so that the vast majority of patients have actually unique neoantigens and not so many shared antigens. But that was kind of somehow expected, because it's rarer to have a shared antigens compared to have random neoantigen or random mutations. And now, just before, just to conclude, I would like to also discuss a bit what are the limitations and some consideration or potential improvement that can be made when it comes to T cell activation, to screening antigens using T cell activation. So this is an example of what was the approach in Rosenberg's group, but this can be also used for other settings, also when it comes to, for example, evaluating recognition to viral antigens or other type of antigens in general. So if one wants to measure to validate, let's say, a target, a T cell target through T cell activation, a lot of different variables needs to be taken into account. A very important aspect is that the T cell status, or like the T cell, for example, exhausted phenotype, both for cancer but also for viral infection, can impact the activation capacity of a T cells. So depending on the readout that is chosen, one may have an underestimation of the actual T cell activation and therefore of the actual potential targets that are present in a certain disease, for example. It also depends on the strength of the stimulus. So a simple way of thinking it is that, let's say you have like a peptide titration. So you can have high concentration of peptide that gives a strong stimulus. But if you go down with the concentration, then the T cells will have less and less stimulation, and then you may not see an activation, even though that is actually the right target, but you just use it at not a suboptimal concentration, for example. And also, as I said, like the readout chosen, it's important, because it could be that if your readout is cytokine secretion, but you only focus on one type of cytokine, maybe you may miss other cytokines produced by other subtypes, and then you may underestimate the reactivity for a certain antigen. So just to summarize, one suggestion is that so whenever it comes to T cell activation and stimulation of T cells, the major danger is to have a suboptimal stimulation, so that you may actually miss a lot of positives. So the best strategy is to actually implement different assays, for example, and use different conditions to be able to cover as much as possible. And with this, I would like just to thank my team at Karolinska Institute, and also the funding agency and collaborators and foundation that make possible my research at Karolinska. And thank you for the attention, and thank you for GenScript again for this opportunity.
Thanks, Anna, for the fascinating presentation. Before we head into our Q&A session, I would like to take this opportunity to share a little bit about GenScript peptide synthesis service. GenScript is a world-leading CRO company with extensive peptide synthesis experience. With our proprietary microwave-assisted PepPower platform, GenScript boasts a strong track record, having synthesized more than 120,000 peptides annually, with an impressive success rate of 98%. In addition to peptide synthesis, we also provide peptide library service with great value to worldwide researchers. In 2020, GenScript has launched a neoantigen peptide synthesis service. We have the technical means and the experience to synthesize large numbers of challenging neoantigen peptides and provide tailored optimization tools for your downstream applications. For more information, please visit our website or email us at peptide@genscript.com. With that, I would like to start our Q&A session, and we'll be happy to take any questions.